क्लास है अभी।
अभी
हाँ।
अ, कल
तो
आऊँ सुबह?
सुबह या तो फिर जल्दी आना पड़ेगा, नहीं तो मैं दोपहर को बुला
दोपहर में आना?
हाँ, दोपहर
ठीक।
में बुला। ठीक।
अभी संडे क्लास?
हाँ, लेना है।
ठीक।
ठीक। So, good evening, good afternoon, guys. Today we'll be starting with week 6 of the course Host Pathogen Interaction by Professor Himanshu Kumar. And in this lecture, we'll be discussing about mostly the recognition receptors. So, in the last class, we have discussed about the toll-like receptors. Okay, so that can recognize various kind of bacteria, fungi, and viruses. And now in this video lecture, we'll be discussing about rig1 like receptors that recognizes basically RNA of the RNA viruses, and the nod like receptors that recognizes wide range of PAMs. Okay, and induce inflammation reactions. And lastly, how these pathogen receptors actually sense the DNA in a cell. Okay, so let's start with the RLRs, that is RIG1 like receptors. Okay, so these receptors actually consist of a family that contains of RIG1 and MDA5. Okay, so these receptors are majorly present in the cytoplasm of the cell that will recognize mainly the RNA and the of the RNA viruses that infect the cell. Okay, so why these are present in the cytoplasm? Because whenever a viral particle enters, so for example, this is a virus that is infecting this particular cell, it will release its genetic material into the cell. Okay, now this genetic material, because it has already been attached to its receptors, so we just now discussed that these RLRs are present in the cytoplasm. So why they are present in the cytoplasm? Because whenever a virus that carries a RNA genome, okay, so this virus, whenever it will infect the particular cell, so what will happen? After attaching to its receptor, it will release its genome into the cytoplasm. Okay. Or the second thing what could be there is it can directly enter into the cell in the form of an endosome, so that the whole endosome will get captured in the cell itself. So now, whenever this kind of thing happens, there are certain receptors in the cytoplasm that are part of the innate immunity, and those will capture or recognize the RNA or the virus that carries that RNA into the cell. Okay, so now these receptors now will these receptors are RIG1, MDA5, LPG2. Okay, so RIG1 stands for retinoic acid inducible gene 1 or DDX58. Then MDA5 stands for melanoma differentiation associated protein 5, and LPG2 stands for Laboratory of Genetics and Physiology 2. Okay, so these are the full forms of these particular receptors. Now, out of these receptors, RIG1 and MDA5 are present in the epithelial cells and the macrophages. Okay, so these are the major sites where the RNA viruses infect. So, this is the place where these two receptors are majorly located inside the body. Talking about LGP2, LGP2 is like a regulator. Okay, so it actually is increased when these two guys activate. So this rig1 and MDA5, once activated, the signaling pathway also increases the LGP2. That increases their effect, and once the effect is done, then it levels actually lowers, so that it can lower the effect of these two receptors. Okay, so it acts as a regulator in effect of these particular receptors. Okay, and they have a similar structure. So, for example, they have a card domain. These two are the card domain. This is a helicase, and this is their CTD, that is C terminal domain. Okay, so helicase is the major site. So, as you can see here, just 1 minute. So, as you can see here, the major site, this is the structure of the rig1, for example. So, helicase is the site where it can easily recognize the double stranded region of the RNA, okay, or any kind of DNA. So, it recognizes the base pairing basically, and the CTD recognizes the endpoint or the phosphate group of the particular the 5 prime phosphate group of the viruses, the free phosphate group. Okay, and where it lacks the methylation. Okay, so basically what happens is there is a term called CPG island. So CPG island is basically recognized by these receptors because this CPG island is very less methylated in the body. Okay, so this is the typical structure and how it recognizes the RNA viruses. Okay, so any doubts still here? Okay, so let's move on. How and what these receptors actually recognize? Okay, so MDA5 basically recognizes encephalomyocarditis virus. Okay, EMCV is recognized by MDA5. So there are these receptors now being distributed into specific viruses that they recognize. So MDA5 recognizes EMCV and poly-IC. Okay, now, the RIG1 recognizes Newcastle disease virus, vesicular stomatitis virus, and Sendai virus. So these are typical viruses that are recognized by the RIG1. And the this region, the card region of the this particular RIG1 recognizes the 5 prime triphosphate end of the RNA. Okay, so because once the RNA is transcribed, so it has a 5 prime triphosphate end, so increase its stability. And the first nucleotide that is added during the transcription. So this is recognized by this particular rig1, and that particularly the card domain. Okay. Now after attaching to these particular viruses, they bind with IPS1, and that leads to the activation of IRF3 or 7 and NF kappa B. So this will increase the production of type 1 interferons and pro-inflammatory cytokines that will actually lead to antiviral response. Okay, so this is how these particular receptors work and how they get activated in response to different viruses. Okay, so these are all the viruses, and not typically the RNA viruses, the DNA viruses also, that leads to their activation. So that leads to their activation. For example, the DS double stranded DNA virus. For example, herpes simplex virus, Kaposi sarcoma associated HSV, and then Epstein-Barr virus. They all activate RIG1 and MDA5. Then vaccinia virus, adenovirus, they both activate RIG1, MDA5, and adenovirus specifically activates RIG1 but not MDA5. Okay, then the rotavirus, that is also a double stranded RNA virus that can activate both RIG1 and MDA5. Okay, then EMCV that we just discussed basically activates MDA5 but to some extent RIG1. Then we have Zika virus, hepatitis C virus. They activate both MDA5 and RIG1. Coronavirus that we just now saw in a couple of years back can also activate RIG1 and MDA5. That is also a single-stranded RNA virus with a positive sense or the sense genome. Then the negative sense genome, influenza A virus, typically activates RIG1. Measles, Ebola, both of them activate both MDA5 as well as RIG1. Okay, the single-stranded RNA virus that requires its expression by the reverse transcriptase, for example HIV, it can also activate both once recognized. Okay, so and the hepatitis B virus, that is a double-stranded DNA virus, that also can be recognized by both RIG1 and the MDA5. So these are some of the typical examples that have been studied in literature that they are known to activate both RIG1 and MDA5. Okay, so moving on to these particular RLRs and how they have a role in the recognition as well as the therapeutic in the therapy perspective. Okay, so in the literature, it has been studied that RIG1 immunoprecipitations after HSV, that is human herpes simplex virus infection, highly enriched and particularly the 5S RNA pseudogene transcripts were found to be bound to RIG1. Okay. Then host cellular RNAs that are unusual, misfolded, or mislocalized or misprocessed are also recognized by RIG1. Okay. So what happens is this is not only with the outside viruses or the viruses that infect humans. This is in response to any of the uh, any of the misfolded, misprocessed, or unusual RNAs that is present in the body are recognized by RIG1. Okay, so that will be a part of the removal of those particular RNAs that are produced variably inside the body. Okay, then disturbances of cellular RNA homeostasis, unmasked cellular non-coding RNA including miRNA. So all the unmasked RNA, miRNA that can be recognized by the RIG1. So mutation in RLRs cause spontaneous signaling in the absence of the virus infection also. Okay, so this is what they are trying to say. Okay, so there are some of the diseases associated with these particular receptors, that is Singleton-Merten syndrome and Aicardi-Goutieres syndrome. So in Aicardi-Goutieres syndrome, the long hairpin derived from inverted repeat ALUs elements have been suggested to activate forms of MDA5 mutation, mutant forms of MDA5 form. So because ALU elements are basically the transposons. So these transposons are jumping genes. Okay, so what happens is whenever these jumping genes are there, they can actually activate some of the silent genes that are present in the body. So the mutations that are present in MDA5 need can be activated by these ALU elements. So that leads to a disease called as Aicardi-Goutieres syndrome. Okay, so this MDA5 mutations lead to irregular signaling of the RLRs, and that can initiate the immune response when the RNA viruses are not present at all in the body also. Okay, then the alteration in RNA metabolism can generate RLR stimulatory RNAs. Okay, so whenever there is a alteration in the RNA metabolism, so there is a metabolism that takes place of the nucleic acid also. So whenever there is a alteration, they can generate the RLR stimulatory RNA, so that they can actually overcome or regulate that particular metabolism. Then the MDA5 also get activated whenever there is mislocalization of the mitochondrial DNA. Okay, so as we have seen, oh sorry, mitochondrial RNA, because the mitochondria, because of the endosymbiont theory, it is considered that it is not actually a part of the animal cell. So it is originated from a bacterial cell. So that's why the mitochondrial RNA, although it will be produced by the particular mitochondria present inside the cell, but the location of that mitochondrial RNA, if it is present in the cytoplasm, then it will lead to the MDA5 activation. Okay, so this is how the MDA5 and the RIG1 actually can initiate their response in absence of the RNA virus also. So that these responses are basically due to the these viruses are basically due to the activation of the receptors in absence, or the it includes the regulation of the different RNA particles present inside the cytoplasm. Okay, so then adenosine deaminase acting or RNA1 deficiency in humans and mice causes chronic type 1 interferon production and interferon in inflammatory pathology. Okay, so this also is associated with the RIG1 and MDA5 signaling as well. Then, in breast cancer, the tumor microenvironment results in the production of the non-coding 5' triple phosphate containing RNA in the stromal cells. So, uh, because of this non-coding RNA, what will happen? It will happen the it will transfer the RNA through exosomes to cancer cell, and that will activate the RIG1 activation. Okay, so these are some of the examples where it can lead to some of the diseases because of the activation of irregular RNAs that are produced from our body. Okay, so any doubts in this particular slide? Okay, if not, then we'll move on to the next topic, that is how do these RLRs actually signal. Okay, so as I told you, whenever an virus enters or it tries to enter into the body or a cell, it releases its RNA. That RNA can be recognized by RIG1 or MDA5. Okay. Now, after binding to that RNA, it leads to a protein called as MAVS, that is mitochondrial antiviral signaling protein, or the MAVS. Okay, so this MAVS also has a card domain and a transmembrane domain. Okay, so now this MAVS targets peroxisome, this targets peroxisome, this targets endoplasmic reticulum and mitochondria by just so this actually is present on peroxisome, my endoplasmic reticulum, and the mitochondria, and it gets activated upon activation of the MDA5 or RIG1. Okay, now it activates a inflammatory pathway. That means either TBK1 or IKK epsilon, that will activate IRF3, 7 or NF kappa B. Now that will activate the IFN gamma interferon uh, interferon producing genes, and well as a inflammatory response. Okay, so this is how the antiviral response actually happens in the presence of the RLRs. Okay, so these are the proteins that are the signaling proteins that are activated by the RLRs during the infection through a virus. Now, moving on to the signaling in a particular uh, more deep. So, there are certain viruses, there are certain microRNAs or non-coding RNAs that actually regulate this signaling. Okay, so what happens is, this is basically the RIG1 and the MDA5, and there are certain modifications that regulate this signaling. So once these uh, these recognize the viruses, there are certain microRNAs like microRNA 576A and LNC RNAs that inhibits the role of the site different components of these particular uh, these particular domains, so that they can inhibit their activation of the signaling, so and that will not lead to the antiviral response. Okay, so I'm not going into that much detail because that will be too much knowledge for now. And but these kind of regulations takes place inside the body, so that the immune response is actually limited to a certain extent only. Okay, the same thing happens with the post translation modifications that can be activating as well as inhibitory in terms of the immune response. Okay, so for example, the activating PTMs are ubiquitination, then ubiquitination and sulfonation. So these two are activating PTMs, and the inhibitory PTMs are phosphorylation and the ubiquitination of the card domains at lysine 115 and the 146. So their specific location at which the PTMs act are the major point that will lead to the activation or inhibition of the particular domains or the particular protein. Same way, so this we talked about the RIG1, and same in the same way the MDA5. Some of the domains are ubiquitinated for its activation, while the others are ubiquitinated for their inhibition. Okay, so same way for the phosphorylation and sulfonation also. So this is how the activation and the regulation basically of the RLR signaling takes place. Okay, so I'm this is actually out of scope. Discussing this is out of scope of the study, but I just introduced these things so that you guys will actually know how this signaling actually is complex, and how different molecules inside the body actually regulates its activation as well as inhibition. Okay, so now moving on to how these these receptors can be used for the therapeutic purposes. Okay, so for example, the RLRs agonists. So agonists are basically those molecules that can activate the receptors. Okay, so these comprise of poly IC, and that will have a 5' triphosphate double stranded RNA or a stem loop RNA. stem loop RNA 14, and M8. So these are the agonists that can activate RLRs without any direct reaction with the RNA viruses or the RNA particles that are present in our body. Okay, so what will happen if these agonists are given? So they can induce cancer cell apoptosis and anti-tumor immune response. So basically, these can be used as an anti-tumor therapy. Okay, so whenever we need a therapy for a specific type of tumor, we can inject these directly into the tumor, or we can direct those to the tumors in terms of nanoparticles or any other type of gene therapy, so that they can be directed to the tumor and they can initiate this and apoptosis and the anti-tumor immune response. Okay, then M8 Rig1 agonist, it is a uridine-rich hairpin 5' triple phosphate RNA that is composed of 99 nucleotides that induces immunogenic cell death through damage-associated molecular pattern or the DAMPs from the tumor cell. Okay, so that means this RNA is composed of a 5' triple phosphate RNA that is composed of 99 nucleotides that is related to these tumor cells. Okay, so these tumor cells would have certain patterns that would make them pathogenic. Okay, so now after recognizing those particular dams, if we give this M8 to the particular tumor, then it can easily activate rig1, so that will lead to anti-tumor immunity. Then we have another SLR14, that is stem loop RNA 14. This act as a powerful immune adjuvant and induced robust anti-tumor response. So adjuvants are basically those molecules that can increase the immune response. Okay, so adjuvants are basically given so that in terms of production, these actually increase the immune response as a side molecules. So in response to any antibodies or something, wherever we have to increase the potency of that particular antibody, we give adjuvant so that we can increase the immune response. And these SLR14s are used to induce robust anti-tumor responses. Okay, then a nanoparticle that can deliver Rig1's natural ligand triple phosphate double stranded RNA into pancreatic cell successfully ceased tumor progression. So this is a study in which when a nanoparticle mediated delivery of Rig1's natural ligand, that is a double stranded RNA that have an extended triple phosphate 5' triple phosphate, that into the pancreatic cell cancer directly, that helped in seizing the tumor progression. So that can lead to activation of the this pattern recognition receptor RLR's pathway, and that can reduce the anti-tumor tumor progression inside the pancreatic cell. Then another example was co-treatment with poly-IC, that is high molecular weight, and ionizing radiation enhances the anti-cancer effect and the following upregulated fast expression in response to fast cell uh, induced apoptosis. So in terms of cancer cell, what we see is we have to target the apoptosis so that we can increase the apoptosis in the cancer cells. Okay, so another type of apoptosis is the fast L ligand apoptosis. So this is a type of apoptosis. There are multiple pathways through which apoptosis takes place. Now we can target different pathways in different type of cancers. Okay, so whenever we can give the co-treatment of high molecular weight poly-IC, that is also an agonist of RLR, along with ionizing radiation, so that actually upregulated the fast expression and induce the fast L, that is fast ligand apoptosis. Okay, then in another example, the RIG1 agonist SLR20, so that is the stem loop RNA 20, can induce immunogenic cell death and leading to inhibition of the tumor growth and metastasis. So this is one of the other examples. Sorry. Now moving on to how these increased immune response can lead to some of the inflammatory responses in the body. So if there is prolonged exposure of type 1 interferon environment, this leads to an anti-inflammatory response, and in inappropriate use of RLR's agonist, this result in out of control inflammatory responses. Then high RIG1 expression is associated with the ovarian cancer. So whenever there is a high expression of RIG1, so that is also a receptor, but that is associated with the ovarian cancer. Okay. So whether there are some of the positive sides, there are also some of the negative sides that lead to some of the diseases inside the body. Okay. So if you guys have any doubt, you can ask. Okay. Now we'll move on to the third type of receptors, that are nod-like receptor or the NLRs. Okay. So NLRs were first identified in plants. Okay, and as a plant resistant genes. So those are called as R genes in plant, and they had a critical function against microbial and parasitic pathogens. Okay, so now they are termed as NLRs. Homologous to the NLRs are present in vertebrates and primitive organisms such as sea urchins, and the evolutionary conserved NLRs family play an important role in function in host defense. So they have a primary role in the host defense. Okay, so that means in the immune system. So in humans, the NLR family is composed of 22 proteins. So the family itself comprises of 22 proteins, but in case of mice, there are 33 NLR genes. So that means in mice, comparatively, there will be more number of NLRs as compared to humans. Okay, so these are some of the example of the NLRs that are present in the body, starting from C, C2TA, N41P, then NOD receptors, NLRC345. Then we have the NLRP family that extends from 1 to 40. Okay, then we have NLRX1. So these are different types of proteins, NLRs that are present in the particular cells. Okay, so then we have the structure of these NLRs are basically similar in all respect, but there can be some variations based upon the particular role and the particular location that it has been produced. So the basic thing is it has a card domain, a AD domain, a nod like receptor, the receptor protein for the disc, for the for the particular pathogen to be accepted, and the LRR domain. Okay, so these are the domains that are typically present in all the NLRs, but some of the domains can be extra. For example, in NOD1 there are two card domains. Okay, and in some of the unspecified locations there can be either a card domain or an extra NOT domain present. Okay, so this is how there are different structures of different NLRs that are present in the body. Okay, so I am not going into that much detail what function and what is the other name of the particular NLRs, but the major thing is you have to just keep in mind that most of the NLRs have similar function, similar structure, but they recognize different bands. Okay, so let's move on to the major structure of the major NLRs. So, for example, these are the node 1 and node 2, then NLRP1, NLRP3, and NAIP5. So, node 1 and node 2. Node 1 basically can recognize IEDAP. So, this is a part of the gram positive bacteria cell wall. Okay, then Listeria, monocysteine, and so on and so forth. So these are the different bacteria that can be recognized by NOD1, and they induce production of these inflammatory cytokines through NF-kappa B and MAP kinases. Okay, then the NOD2, MDP, Mycobacterium tuberculosis, Listeria, monocysteines, and Streptococcus pneumoniae activate NOD2, and they also induce the production of inflammatory cytokines through NF-kappa B and MAP kinases. So this is how they function in the particular cytoplasm. Then talking about NLRP1, so MDP lethal toxin of Bacillus anthracis, they induce caspase 1 dependent cell death. So whenever a cell is infected by the viruses and NLRP1 recognizes whether the particular cell is infected or not, then it can induce the caspase 1 independent cell death in those particular cells. Then we have the NLRP3. So NLRP3 is a major component that recognizes MDP, microbial RNA, these ligands from different toxins, then UV radiation, beta amyloid protein, picryl chloride. All these are the ligands for NLRP3, and they induce caspase 1 dependent IL-1 beta and IL-18 production. Then they form the NLRP3 inflammasome along with ASC and caspase 1. Okay, then NAIP5, that is induced by Legionella pneumonella. So these control replication of bacteria independent of caspase. Then we also have another NLR that can induce caspase 1 independent IL-1 beta and IL-18 production. Okay, so this is a typical how they. So most of the NLRs either use caspase 1 dependent cell death or caspase 1 independent cell death. So this is how both the NLRs, both type of NLRs function. Okay. So any doubts in this particular slide? Okay. So let's move on. So like RLRs also signal in some of the ways. So they actually can activate the NF-kappa B pathway. Now NLRs also activate the NF-kappa B pathway, but in a different way. Okay, so then we have the NOD1 and NOD2 receptors. So these receptors can activate through through cardiac, and this cardiac can directly activate NF-kappa B in response to P50 and P65. So those are also proteins that activates the inflammatory response genes. Okay, so and then they can activate card9, MAP kinase, and then the inflammatory cytokine. So MAP kinase is one of the important pathway through which the inflammatory cytokines are produced. Okay, so they have both the conditions. They can directly activate NF kappa B through P50 and P65, or they can activate MAP kinase through the inflammatory cytokines. Okay, so this is how node 1 and node 2 actually signals, and then there are certain inflammasomes, as we said, that they either function in caspase 1 independent or caspase 1 dependent kind of conditions. So we have NALP3 that is activated in response to ligands. Then we have NLRC4 and NAIP5 and NLRP1 that in response to ligands they activate caspase 1 dependent pathways that increases the IL-1 beta family of cytokines, and that can release IL-1 beta and IL-18. Okay, so these two are released by these kind of inflammasomes once they are activated. Okay, so this is how NLR signaling takes place. So there are different type of inflammasomes that can be formed from the different kind of NLRs. Okay, so NLRP1 that can be activated by lethal toxin MDP or ATP that activates caspase 5 and caspase 1 through ASC. Okay, so now this is the type 1 inflammasome. Then we have the NLRP3 inflammasome. So they can be activated through multiple pathogen associated molecular pattern or damage associated molecular patterns. Okay, so PAMPs are basically toxins, bacterial RNA, viral and host nucleic acids, and DAMs including extra ATP, potassium ion efflux, calcium signaling, crystal or aggregates, lysosomal disruption, mitochondrial disruption, ROS, a reactive oxygen species, ceramide, mitochondrial DNA, and cardiolipin. Okay, so these are basically the damage associated molecular patterns released in response to any of the diseases or any of the metabolic changes that takes place inside the cell. So these activate NLRP3 associated inflammasome, that is an important inflammasome, and that to function in caspase 1 mediated activation of the cell death as well as the inflammatory response. So this is type 2. Then we have the NAIP and the NLRC4 dependent inflammatory response that is basically targeted by the flagellin or the rod proteins. And then the cytoplasmic DNA activates AIM2 that also activates the caspase 1 dependent inflammatory response. So these are certain types of inflammatory responses in response to different ligands that the different NLR proteins bind to. Okay, so moving on to how these NLRs actually function in disease as well as the mutations that cause the diseases. For example, NLRP3 mutation, and that is causing the disease. So they cause cryopyrinopathies. That means they have the issues with the skin, eyes, and joints. Okay, then we have the mutation in the NOD2 that causes Blau syndrome or early onset of sarcoidosis. That also is a disease for the skin, eye, and joints. Okay, so now loss of function mutation causes in the NOD1 causes asthma, in node 1 also causes sarcoidosis, that is in lungs, both in lungs. Then in the CTHA causes bare lymphocyte syndrome in the lymphocytes, and both in node 1 and node 2 in intestine can cause inflammatory bowel disease. Okay, and if there is any NLRP1 mutation, that can be gain of function or loss of function, that can cause vitiligo, that is associated with the skin. Okay, so that is associated with the skin. So if any of the mutations in the NLR receptor lead to various diseases. There is also one of the diseases called as Crohn's disease. So that is also an inflammatory disease associated with the NLRs. So this is an intestinal disease. So what happens is, in this case, the reduced not to dependent expression of bactericidal alpha defensins in the Paneth cells. So Paneth cells are the defensive cells of the intestine. Okay, so these cells actually help the intestine to extract the bacteria from the food, and it will lead to the immune response. So whenever there is a less expression of NOD2 from these particular Paneth cells, this leads to impaired recognition and clearance of intestinal phagocytes. Okay, so now what will happen? It will not be able to activate the macrophages for their clearance from the of the intestinal phagocytes. Um, and then what will happen? It dysregulates the TLR2 mediated inflammation in intestinal macrophages and dendritic cells. So the pro-inflammatory cytokines are actually induced, and then what will happen? Because of this, there is enhanced IL-1 beta uh, production, so that will lead to a inflammatory disease of the intestine, that is called as a Crohn's disease. Okay, so this is also one of the example where the regulation of the NLRs can cause a disease. Okay, so any doubts in this particular slide? Okay, so let's move on. So now we are moving on to the DNA sensors. Okay, so how these PRRs also recognize DNA. So it has been known that the DNA sensing is there in through the PRRs that has been reported around 50 years ago. And the first PRR that was used was TLR9, that was reported to detect DNA in the cytoplasm. Now, TLR9 recognizes undermethylated DNA, that is the CpG island, okay, as I stated before. So, this TLR9 basically identifies the undermethylated DNA or less methylated DNA that is present in mostly the microbial genomes. Now, in other examples, we have the tank binding kinase or the TBK1 that is required for the IFN beta production in response to transfected synthetic double stranded DNA or poly DADT. Okay, then we have, after activation of the upstream PRR signaling, TBK1 directly phosphorylate IRF3 that leads to IFN beta induction, and then the signaling access initiates. Okay, in response to the fundamental cytoplasmic DNA, cytosolic DNA. Okay, then we have more DNA sensors that could directly bind to cytosolic DNA and mediate IFN1 induction eh, via a signaling pathway with TBK1 and the IRF3. Okay, similarly, so now second this thing was TBK1, tank binding kinase. The third one was, first reported cytosolic DNA receptor was identified as activator of the IRFs, that is DIA. So DAI. So DI or ZBP1, sorry. So DI or ZBP1, it encodes also, is it encoded by an IFN inducible gene that was found to be capable of expression upregulating the expression of IFN1 via NF-kappa B and IRF3 response to poly DAD. So the second DNA sensor is DAI or ZBP1. Okay, then the RNA polymerase 3 that is capable of transcribing 80 rich double stranded DNA can produce a RNA moiety that contains 50 triphosphate moiety that is recognized by RIG1, leading to IFN beta induction. no role, then in no role of DAI in macrophage responses to DNA was observed, while the in vivo response of DNA vaccination was TBK1 dependent but DAI independent. Okay, so this is how different DNA sensors were produced, and then another signaling protein, that is STING, also called as MITA, MPYS, or ERIS, that also was recognized as a DNA sensor and that can also directly activate TBK1 via IRF3. Similarly, there are different TBKs, there are different DNA sensors that have been identified since till now, so that can actually that can actually detect the DNA in the cytoplasm. Okay, so this DNA can be either the DNA of the pathogen or as the any dysregulated DNA that is present irrespective of the self DNA. It can be detected by these proteins. Okay, so now we'll go a bit deep into the signaling and how different DNA sensors actually recognizes these particular DNA. So we have a TLR9 and the DNase complex that recognizes those DNA and they cleave those DNAs. Okay, so DNase 1 cleaves single stranded DNA and double stranded DNA in extracellular space, and the cytosolic DNA sensors are CGAS, IF116, DDX41, AIM2, PQBP1, and DA1. And then we have the nuclear DNA sensors. These are also IF116, BRCA1, RAD50, MRE11, and DNA-PK. Okay. So, and DNase2 along with TLR9, these senses both extracellular DNA that is translocated in the endolysosome. So this is how TLR9 in combination with DNase1 can recognize the particular DNA molecules. Now we'll see how TLR9 actually recognizes the CpG DNA in the endolysosomes. Okay, so the TLR9 is a monomer. Okay, so this is a TLR9 molecule that is a monomer. Now, whenever it sees a CpG island that is present in the endolysosome, it will dimerize. So it dimerizes. Now it activates MYD88. That is a major signaling molecule in the TLR signaling. So now it will activate MYD88. Now this MYD88 activates TRAP3, IRF7, IK, the NF kappa B pathway through P50 and P65, and they will release the type 1 interferons and the pro-inflammatory cytokines. Okay, and the important thing is that the binding of TLR9 to the CpG DNA is pH dependent. Okay, because the endolysosome pH is nearly 4. Okay, so this is how the binding and the activation of the inflammatory pathway through TLR9 takes place. Now, TLR9 has been reported or has been seen in the literature that it can easily detect the DNA of these particular viruses and the bacteria and the fungal molecule also. Okay, so there are some of the viruses that are mentioned here. Then we have the Neisseria meningitis, so and Mycobacterium tuberculosis, Aspergillus fumigatus. So these are the fungal, bacterial, even the parasitic molecule also, and the rest are the viruses that has been shown to be detected by the TLR9. Okay, so now there would be some of the pathways that how the DNA sensing takes place. So I'll not go into that much detail, but the major pathway that takes place is through IRF3 and the TBK1 that activates the type 1 interferon genes. Okay, so this is the major pathway that helps in the production of the type 1 interferon genes, and the second pathway that is activated by is P50 and P65 mediated production of NF-κB signaling, and that leads to the pro-inflammatory cytokines. Okay, so this is cyclic GMP AMP synthase. So these also can lead to the production of a cyclic GMP. Okay, so cyclic GMP. So this leads to binding to a sting, that is an endoplasmic reticulum adapter. So now this adapter leads to the production of the type 1 interferon gene by the IRF3 and TBK1 pathway, and the pro-inflammatory cytokines through NF-kappa B pathway. Okay, then in another example, we have the immune sensing of cytoplasmic DNA or the double-stranded DNA as non-self nucleic acid. Okay, so this C gas, that is so the cytoplasmic DNA is efficiently degraded by the 3' repair exonuclease 1 TREX1 located in the cytosol. The oxidation of DNA, which occurs in situations of the oxidative stress, leads to oxidation of DNA, which is common of which is 8-hydroxyguanosine, 8-OHG to be precise, and this stabilizes DNA against degradation by TREX1. Okay, so the C-gas system that we last discussed in the last slide, this oxidized DNA is recognized by the C-gas system, and whenever there is high amount of oxidation, that oxidation stress inside the cell, the DNA, the guanosine is basically oxidized to 8 hydroxyguanosine. So this is because of the oxidation. So now what will happen, the trex basically recognizes this 8 OHG, and it will lead to its so it actually recognizes the 8 OHG, and it helps in stabilizing against it degradation. Okay, and if there is accumulation of this oxidized DNA, that will lead to the production, that will lead to the recognition by C gas, and that will lead to type 1 interferon production. Okay, so this is how the oxidative stress related DNA is also sensed by C gas. Okay, now we have another DNA sensor, that is AIM2. So it reacts with the ASC protein that leads to an inflammasome production. Okay, so now this is a slightly different pathway where AIM2 leads to a caspase 1 dependent inflammasome. Okay, so this can lead to the production of IL-1 beta and IL-18. So these are also inflammatory cytokines, as well as it can lead to the production of gas germin D. So that leads to pyroptosis. Okay, so this is how different DNA sensors work. So another sensor that we talked about is IF116. So this is present both in the cytoplasm as well as in the DNA. Now, as well as in the nucleus. So once they actually recognize the IF116 recognizes the DNA, it leads to the same common pathway for the type 1 interferon production, that is the IRF3 and the TBK1. Okay, so this is how different DNA sensors function. So if you guys have any doubt till now, you can ask. I didn't cover that much in detail because the major pathway that they cover are the same. Just the how and what they recognize is the major difference. Okay, if no doubts, then we'll start with the question. So, what makes LGP2 different from other RLR family members? So, what can be the correct answer? So, yes, the correct answer is card domain. So, the RIG1 and the MAD5 both have a card domain, then they have the helicase and the CTD. Okay, so both have the similar structure, but LPG2 do not have a card domain, it has directly the helicase and the CTD. Okay, so there is absence of the card domain. So let's move on to the next question. What role does NF kappa B plays directly in the RLR signaling pathway once activated by RIG1 and MDF5? So what can be the correct answer? Okay, so the correct answer is option 3, that is transcription of the inflammatory cytokines. Okay, so what happens is this NF kappa B is basically a transcription factor that activates other transcription factors that leads to the release of the inflammatory cytokines. Okay, so this is how the NF kappa B takes place. Okay, so let's move on to the next question. Which of the following is a distinct role of IF116 sensor compared to other DNA sensors like AM2 or CGAS? What can be the correct answer? So the correct answer is option number one, that it can sense both DNA and RNA as well. Okay, so IF-116, it is present on both cytoplasm as well as nucleus, and it can sense both DNA as well as RNA as compared to AM2 and CGAS. So CGAS can detect only DNA, and AM2 also can detect DNA. Just the difference was CGAS actually activates through sting, and AM2 activates through caspase 1. Okay, so any doubts in this question? Okay, so let's move on to the next question. What is the key role of loss of function mutation in the NLRs? So what can be the correct answer? Okay, so yes, this is the correct answer. So as we have studied in a few slides back, so the loss of function mutation in the NLRs can lead to immune deficiency syndrome like Barr lymphocytic syndrome. Okay, so let's move on to the next question. Which of the following statements describe the implications of NLR mutations in human diseases? So what can be the correct answer? Okay, so let's see. So the correct answer is 1, 2, 3, and 6. Okay. So NLR mutations can lead to both gain and loss of function diseases. Yes, we have seen this. Then mutations in the NLRP3 are linked to variety of inflammatory diseases. Yes, because NLRP3 is important in forming an inflammasome in response to various damps. Okay, So that's why any mutation in the NLRP3 can lead to variety of inflammatory diseases. Then NOT2 mutation has been associated with Crohn's disease. Okay, so yes, we have seen this. This is also an inflammatory disease of the intestine. Then loss of function mutation in the NLRs will often result in heightened immune response. No, that will be leading to lowering of the immune response. Gain of function mutation in the NLR and into immune deficiency syndrome. No, that will be heightened immune response. Okay, genetic variations in NLR contribute to diverse immune response and disease susceptibility. Yes, this is correct. This can be uh, like true for any of the receptors that are present there. So the genetic variations can lead to disease susceptibility as well as difference in the immune responses. Then NLR mutations exclusively called autoimmune diseases? No. Then all NLR mutation lead to inflammatory conditions? No. Okay, so this is also false. So the correct answers are 1, 2, 3, and 6. Okay, so let's move on to the next question. Which is not an adapter of our RIG1 protein for downstream signaling? So what can be the correct answer? Excuse. So downstream means that the signaling that takes place from one of the molecules. For example, RIG1 will activate some of the molecules, so that will lead to the activation of other molecules following the following the production of the inflammatory cytokines. So likewise, downstream means like that. Okay. So here the correct answer is option 4, that is TRAF6. Yes. Okay, so it can activate RIG1 activates MAVS, IPS1, Cardiff, and Visa, but it will not activate TRAF6. No, it also activates IPS1, okay, RIG1. TRAF6 is not an adapter of the RIG1. Okay, okay. So let's move on to the next question. Which of the following statements are true about activation process of RIG1 and MDA5 in the RLR pathway? So what can be the correct answer? Just one. Yeah. Okay. Any more answers? Okay. 7 also. Okay, so let's see the answer. So the correct ones are 3, 5, and 7. Okay, so uh, let's see the options. They are always in active form? No. The activation occurs in the peroxisomes? No. Not in the peroxisomes, basically. Their activation occurs in peroxisomes, ERs, and then the mitochondria. So these are the major organelles that help in activation. Then activation leads to conformational changes exposing card and helicase domains. So yes, this is correct. So as we have seen that they have the card and the helicase domains. So the conformational changes leads to their uh, the activation leads to the conformational changes in these domains. Okay. Activation involves phosphorylation of MABS? No. Okay. So it doesn't phosphorylate, it actually changes its conformation. They recruit adapter molecules with card domains for signaling? No. Only Rig1, not MDA5 activates NF-kappa B? No, both of them activate NF-kappa B. Their interaction with viral RNA initiates the activation process. Yes, this is correct. They are permanently anchored to the mitochondrial membrane. No, they are present in the cytoplasm. Okay, but the MAVS that is present on the mitochondrial membrane. Okay, so the correct answer is 3, 5, and 7. Okay, so these are the correct answers. Okay, so let's move on to the next question. Which of the following statements best describe the function of ASC in the inflammasome complex? So what can be the correct answer? Okay. Any more answers? Okay. So yes, the correct answer is option number two. So ASC is basically apoptosis associated spec-like protein. Okay, so this is an adapter molecule that is attached to the that is essential for activating the inflammasome complex. Okay, and it actually activates through card card interaction. Okay, so it also has a card domain that interacts with the card domain of the caspase 1. So it recruits caspase 1 that cleaves into the pro caspase 1 that cleaves into the caspase 1, and then it leads to the activation of the inflammasome complex. Okay, so this is how the function ASC functions. Okay, so let's move on to the next question. In the context of DNA sensing and type 1 interferon production, what is the role of IRF3 primarily play? So what can be the correct answer? Okay. Yes, the correct answer is option 4. So what happens is whenever there is an signal through sting pathway, so it actually activates TBK1, which phosphorylates the IRF3. So now this IRF3 is a transcription factor that leads to IFN genes production. Okay, so the correct answer is option number 4. Okay, so let's move on to the next question. Which protein is characterized as an adapter that engages TBK1 upon DNA stimulation? So what can be the correct answer? Okay, so the correct answer is option 2, that is MITA. Okay, so MITA is basically sting. Okay, so sting is the adapter for TBK1. Okay, so this is how the DNA signaling or the DNA recognition takes place. Okay, so let's move on to the that will be IFI, I guess IFI 16. So this was mistakenly kept as exclamation. So this will be IFI 16. So that is also a sensor for DNA that acts on both cytoplasmic as well as the nuclear membrane receptor. Okay, so let's move on to the next question. Which of the following disorders is associated with the mutations in the NLRP3? So what can be the correct answer? So yes, both Muckle-Welch syndrome and FCAS are caused due to the mutation in the NLRP3. Okay, so the Aicardi-Gouttiere syndrome is because of the mutation in the RLRs. Okay, so let's move on to the next question. Which of the following recognizes large double-stranded DNA structures lacking the 2' O methylation generated in the cytosol during virus infection? So what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option number 5, that is MDA5. So yes, MDA5 directly recognizes the double stranded RNA. Okay, so that lacks 2' O methylation generated in the cytosol during viral infection. Okay, so let's move on to the next question. IPS1 or MABS, an adapter involved in RLR signaling, is predominantly located in which of the following answers are correct? So what happens is where in the 2' region. Okay, so we have 3' OH in the DNA, right? So for example, this is the DNA molecule. This is first, second, third, fourth, and here the C. This is fifth. So this is a ribose structure. On the fifth position, we attach the phosphate. Okay, the 3' OH is involved in the phosphodiester linkage. The 2' OH is free. So now 2O is methylated. Okay, so this methylation has different roles. Whenever a viral RNA enters into the cytoplasm, this methylation actually helps in recognizing that it can be a RNA that can be from a outside source. Likewise, okay. No, it can happen in any variable conditions, like because the RIG1 and MDA5 actually activates different in under different conditions also. Okay, and in terms of in the nucleus itself, it is a major modification that takes place, epigenetic modifications for activating or inactivating any part of the gene. So, what can be the correct answer of this question? Okay, so the correct answer is option 4, that is mitochondria. Okay, because it activates mitochondrial associated inflammatory response and cell death. Okay, so the MBAS and IPS are located on the mitochondria. Okay, so let's move on to the next question. Which of the following statements are wrong? So, what can be the correct answer? Mhm. Okay, so the correct answers are 3 and 4. So let's see the options. High RIG1 expression is associated with the ovarian cancer. Yes, we have studied this. Then prolonged exposure to a type 1 interferon environment can lead to an anti-inflammatory response. Okay, yes, this is also correct. Poly-IC as in IC is an RLR antagonist. No, it is an agonist of RLR. So this is wrong. Sting directly engages with MAVS to direct IRF3 activation. No, it engages with which one? TBK1. Okay, so to direct IRF3 activation. Then cytoplasmic DNA is degraded by 3' repair exonuclease 1, that is TREX1. Yes, this is also correct. IF1, IFI16 and ZBP1 DA1 can sense viral DNA as well as RNA. Yes, this is also correct. So the two options that are incorrect are 3 and 4. Okay. So let's move on to the next question. Which of the following statements are correct? So what can be the correct answer? Mhm. So both, uh, yeah, let me check once. Yeah, it will not be able to detect the RNA. So IF-116 do not detect RNA. Okay, but ZBP1 actually can detect both DNA as well as RNA. So option 6 is also wrong. Okay, so yes, thanks for the correction. Okay, okay, okay. So the correct answers are 3, 4, and 5. Okay, so let's see. DNA is 2 cleaves as single-stranded DNA, dsDNA in extracellular space. Yes, this is correct. DNase1 senses extracellular DNA translocated in the endolysosome. Yes, this is all correct. Sorry, these two are false. Okay, so DNase1 cleaves both single-stranded and double-stranded DNA in extracellular space, but DNase2 can recognize the translocated endonuclease along with TLR9. Okay, the oxidized DNA is recognized by CGAS. Yes, this is correct. NLRs are first identified in a ligand for NOT2. Yes, this is also correct. Then MDP is a ligand for NOT2. This is also correct. Blau syndrome is a disorder associated with mutations in NOD1. No, this is false. Okay. So the correct statements are 3, 4, and 5. Okay. So this was the last question for today. If you guys have any doubt, you can ask. So so not to basically, so in terms of not to the NLRs, firstly, so this is basically the term that NLRs, once they were identified, the not to was the first one. Okay, so that's the not to, that was the first one studied, and then the uh its ligands were also studied. Okay, so this is what the meaning of that statement. Okay, if no doubts, no further doubts, then we'll close the session for today. Thank you guys for joining in. We'll meet next week.